So basically, there are two kidneys. Each one has artery and a vein, renal artery and a renal vein that supplies blood for the kidney to function, and also the blood that the kidney actually filters because that's its main function. Then each kidney has a tube called ureter, and ureter sits right here, it's kind of pink. And it goes to the urinary bladder, and urinary bladder you can see right here. This is the male version of this model, but the female version it looks similar, except for some reason they made urinary bladder smaller. It's not smaller in females, it's just probably like this one is full, another one is empty. So, and then from urinary bladder, the urine will go into the urethra. You can see urethra here again, and basically, who put people don't know. Unless it's like, actually somebody put it in the stomach before, but anyway. So that's urethra, and obviously in the male, it's different because in the male, urethra is used for both reproductive and urinary systems. So we have prostate and you have basically ejaculatory duct connect to it. But the female urinary system and reproductive, they do not share parts. They are only close anatomically, but on the body, but not sharing parts. Also, in males, urethra is longer because it goes through the penis. In the females, it's a little bit shorter. So essentially, that's like all the gross anatomy of the urinary system with organs. So documenting pictures of both male and female versions for that. Then we have this model of the kidney. So obviously, this is larger than the kidney in reality in humans. Maybe, I don't know, this may be like an elephant size or something. But basically, what are the parts? In the body, the kidney is also covered by a layer of fat, which you may have seen or will see when we look at CAT, in a couple of labs. I think this lab, then we have another one, urinalysis, and then we have CAT next week, Thursday. So, but anyway, there's an adipose tissue capsule, which will be like here. It protects the kidney physically because it's like partially covered by the ribs, but there is an area where it's not covered by the ribs, and if something hits you in the lower back, you can injure it, but the fat protects it. Then we have the actual fibrous capsule. In this case, it's this, dark purple, or not purple, what is it, maroon color. So this color, basically, that's the actual capsule part of the organ. Adipose is more like just surrounding it. And when you we'll dissect the pig kidney actually, and you can peel off the membrane on the cat as well. We can like peel off this capsule membrane. So, and then inside, you can see the inside structure of the kidney here. It's got two main layers. We have cortex and the medulla. Also, we have these openings. So, but on the outside, you can see this is the ureter. So it starts wide and then becomes a little narrow, but it's a pretty large tube. And of course, you have the artery, renal artery, and renal vein. As always, artery is a little bit smaller because they have higher pressure and the blood goes at higher speed. So artery is a little smaller than the vein. So what is inside them? The cortex is this area, and cortex you have like in different organs. We saw in the thymus and other. It just means a surface layer, like adrenals, surface layer of the organ. Medulla is the middle layer. In the cortex, we have these little structures called glomerulus. We will see them on that board model. Look at the details, but they are microscopic. You have about all glomerulus and nephrons, part of glomerulus, part of nephrons. We have about 1 million of nephrons per kidney. Nephrons are not a single cell; it's multicellular, but very small structures. And nephron does like everything that the kidney can do. If you understand how one nephron works, that's basically you just have 1 million of them, but they work the same way. So we have like 2 million because you have 2 kidneys. But to survive, you need approximately 30%, and that's why a person with healthy kidney is able to donate 1 kidney and still be okay, even though they, of course, will be less healthy because if you lose nephrons, then you will get basically a problem sooner if you have only 1 kidney. But anyway, in the medulla, we have these pyramids of the medulla, and you see them back they're like this. There are several pyramids in the kidney, probably between, I don't know, 6 and 10 or something like this. 1, 2, 3, 4, 5, 6 here, more maybe from the other side. So, pyramids is where the urine is concentrated. It comes out through this tip called papilla, and it goes into the opening called minor calyx. Each pyramid has its own minor calyx that combines several minor calyces. The calyx is one, calyces is plural. They're combined into major calyx right here. So major calyces, there are about 2 or 3 per kidney. And from here, the urine goes into the renal pelvis. It's this area right here. And from there, the urine will flow or be propelled, pushed by the waves in this ureter to the bladder. We also see on this model, we see the branching of the blood vessels. First, the artery and the vein the same way. They branch into these lobar arteries, like the lobes. Then you have segmental arteries. Then you have like these interlobar arteries. And then you have arcuate arteries, like an arc like this, make an arc. And then radiate arteries go like this, so these are radiate arteries. And the veins are the same way. And from these radiate arteries, there are small arterioles that go to each individual nephrons. I mean, small means microscopic, you can see them on the slide. Anyway, so basically, that's the model of the kidney. One other thing that you can see well on this one is basically the hilum. Hilum is not a thing, but it's like the region. It's this region where the tubes are connected. So hilum is present on different organs. Like for example, lung has a hilum, like this area where the arteries and the bronchi connect to the lung. So also, like spleen has it. So basically, just like this area. So now let's look at the more detailed model, which is this one. This model is kind of like a kind of like going from large to smaller, kind of like zooming in. So in this case, you see the kidney, which is very similar, essentially, to that, even in size. Ureter, artery, vein, branches of arteries and veins, pyramids. You see the renal pelvis a little bit more closed on this one, and minor calyces, major calyces. Also, this shows you the nephrons, but the way they're shown, they're much bigger, because these should be like 1 million per kidney. Obviously, if you have like this size, they would not fit, you would fit 1 million. But the idea is that a nephron contains corpuscle and glomerulus in the cortex, and then most of the tube goes in the medulla and have collecting ducts. Now, this is a section basically of something like this, zoomed in, and this shows you more detail. So first of all, you got the interlobar, so this is a lobe of the kidney, capsule, cortex, medulla. We have interlobar artery, goes like this, and makes the arcuate artery like an arc between the cortex and the medulla, and you have radiate arteries coming up. From here, blood goes into this afferent arterioles that each goes to individual nephron. It goes into the blood goes to glomerulus. Then we have, I won't ask you these little arteries, it's very hard to kind of identify. Or maybe, I mean, we can identify them, but usually ask you larger ones. So we have afferent artery and efferent artery. I mean, efferent arteriole. Efferent arteriole makes another capillary bed, peritubular capillaries, like here and these ones. And then blood goes into the venules and arcuate vein, interlobar vein, and down to larger veins. But as far as nephrons, this shows you 2 nephrons. One has a shorter loop, a so-called cortical nephron. There are 2 types. Most of them are like this short loop, cortical nephrons, like 80%, and about 20% with a long loop called juxtamedullary nephrons. You can see how it's spelled. It's basically a nephron that's used for concentrating urine when you need more concentrated. And basically, from corpuscle, the urine or the filtrate, initial filtrate, flows into this proximal convoluted tubule, PCT, then goes in the loop, comes back to the cortex. Then you have distal convoluted tubule, DCT. And from here, it's already now filtrate becomes more or less urine. It goes to collecting duct, and this is collecting duct. There are multiple collecting ducts here, but a collecting duct is not part of one nephron. It's shared by several nephrons. You can see these connections. For example, this is a nephron connected to this duct. This nephron connected to this duct. So multiple nephrons connected, and then urine will go to the end of the pyramid, and at the end, collecting duct ends. It has an opening. This is the papilla, and drops of urine will come out of here into the minor calyx. Now, this is even more zoomed in version. That's a microscopic structure. It's this circular structure called renal corpuscle. So each nephron has one. And renal corpuscle consists of the capsule made of simple squamous epithelium. And this is called Bowman's capsule, or just a nephron capsule. Inside of that, there are special capillaries. See this capillary bed. This is called glomerulus. These are more porous capillaries called fenestrated capillary. Remember, there are like regular continuous capillaries, which are everywhere. Then you have fenestrated and sinusoidal. Well, these are fenestrated. It's like a middle, but higher permeability than regular ones. And from here, the filtrate, the plasma, with everything in it except the cells and the largest proteins, comes out. So proteins and cells stay in the blood and continue to go back to the bloodstream. But everything else, all the, like water, salts, nutrients, toxins, waste, comes out as filtrate. And that filtrate will flow into the proximal convoluted tubule. It's this one is shown here as cut like that, and it's made of cuboidal epithelium. And now here we will absorb stuff that we don't want to go into urine, for example, nutrients like glucose. And that tubule will go to the loop, comes back, and this is distal convoluted tubule right here. Distal convoluted tubule is not connected directly to the capsule, but it went down, came back, and went here. And you can see this is also made of cuboidal epithelium, but there's one difference between proximal and distal. Proximal has microvilli, so it's cuboidal epithelium, but it's fuzzy on top. Because microvilli, just like in digestive tract, it helps absorption. In this case, it's called reabsorption. Because in digestive system, when you eat food, you absorb it like for the first time, right? But here we have the nutrients, we already absorbed them before, but now we filter it, we have to reabsorb them, like absorb them second time. So we don't waste nutrients like glucose should not go to urine, for example. And you can see on the cross section cuboidal with the fuzzy surface microvilli. We'll see that on the slides as well. You saw slides before in ANP1, but we did not distinguish like proximal and distal. We just said that's an example of cuboidal. Now we will have a little more details on that. And also other thing here that you need to know is this tubule. See, this is proximal. Afferent arteriole. Afferent A stands for approach, and efferent arteriole E stands for exit. So blood goes in through afferent, comes out through efferent. If you see that afferent is thicker, the reason is that it basically some fluid will go into the filtrate, so the volume of blood would be about 20% less in the efferent than in the afferent. Also, that that creates higher pressure because the tube that goes in is bigger, the one that comes out is smaller. That creates the pressure that helps the filtration. Also, if you look at this glomerulus, you see it looks a little strange because on one side it's yellow, on the other side it's red, right? The reason is, on one side it shows you these additional cells that cover the capillaries. These are called podocytes. They are present on both sides, but on this side they remove them to show you little tiny holes. You can see them. This is those windows, fenestra or fenestrated epithelium, improve the filtration. But podocytes, they're one of the functions to prevent filtration of like large stuff like cells and especially proteins, because you don't want them in the urine, you want them to keep them in the blood. Anyway, so that's all for this model. So identify the parts and take pictures.